What's up guys, it's the Bio Chef here. Today I'm going to be talking about the fluid mosaic model. So what is the fluid mosaic model? The fluid mosaic model is a biological theory that describes the structure of biological membranes. It proposes that biological membranes are composed of fluid mixture of phospholipids, proteins, and cholesterol. Overall, the fluid mosaic model helps us understand how cells are built and how they work. So let's look at one of the main components, the phospholipid. Phospholipids play a crucial role in the fluid mosaic model of biological membranes as they form the basic structure of the membrane. A phospholipid has a hydrophilic head, which means that it is water-loving. The phospholipid also has a hydrophobic tail, and this hydrophobic tail means that it is water-hating, which means it does not like water at all. It's also important to remember that the hydrophilic head has a phosphate group, and the hydrophobic tail is a fatty group, or fatty acid. A lot of people call them fatty acid tails in this instance. Cholesterol molecules are interspersed between the phospholipids, which help them maintain the fluidity of the membrane and regulate its properties. The presence of cholesterol affects the physical properties of the membrane, such as its stability and its fluidity, which helps regulate the function of the membrane proteins. The membrane proteins you need to know for the fluid mosaic model are the glycoproteins, integral proteins, and peripheral proteins. So let's look at glycoproteins. What is a glycoprotein? It's basically a protein, such as the one you see right here, attached with a carbohydrate, like this one. The way that I like to think of this is think of two pieces of bread. Like, that makes a sandwich, right? What do you usually put between a sandwich? Meat, chicken. Chicken is a protein and a bread is carbs. It's a carbohydrate. So just think of those two things and think glycoprotein. This is how a glycoprotein looks like in the fluid mosaic model. As you can see, the pink circle is the protein and the yellow things that are attached to it are the carbohydrates. The carbohydrates are attached to the protein by covalent bonds through a process called glycosidic linkage. The next protein we'll be looking at are integral proteins. Integral proteins are embedded within the lipid bilayer of a biological membrane. They are integral to the membrane because they are covalently attached to the lipids or otherwise tightly bound to the lipid bilayer. Integral proteins are very important in biology as they act as channels or pumps which control the movement of ions and small molecules in and out of the cell. The next protein we'll look at are peripheral proteins, which also play a very important role in the biological membrane. Peripheral proteins are not covalently attached to the lipids, but instead they interact with the hydrophilic head groups of the phospholipids or with the integral proteins in the membrane. So now, when you look at a fluid mosaic model, you should be able to understand all the components of it. You have the carbohydrate, which is attached to the protein, making it glycoprotein. You have the hydrophilic head, which is also a phosphate group, and you have the hydrophobic tails on the other end, which are fatty acid. You have the cholesterol, which is embedded within the phospholipids, and you have the integral proteins, which can act as pumps or channels. You also have the peripheral proteins that are not covalently bonded to the phospholipids. If you have any questions, leave them down below. Thanks for watching. Peace.